So DNA replication in prokaryotes starts from a sequence found on the chromosome called the origin of replication, the point at which the DNA opens up. Before I say the next step, I've indicated the direction of the replication by the arrow above and the 5 and 3 ends of all the molecules. An enzyme called helicase unwinds the DNA by breaking the hydrogenous bonds between the nitrogenous base pairs. As DNA opens up, the replication forks are formed and gets extended bidirectionally as replication proceeds. After helicase, single-stranded binding proteins indicated by the purple dots then coat the single strands of DNA to prevent the DNA from unwinding back into a double helix. DNA gyrase, indicated by the blue dots here, reduces torsional strain and supercoiling, preventing DNA breakage during DNA unwinding. After that, primase then synthesizes short RNA primers to allow DNA synthesis to begin. After this, DNA polymerase III comes into play. It is able to add nucleotides only in the 5' to 3' direction. It also requires a free 3' OH group to which it can add nucleotides by forming a phosphodiester bond between 3' OH end and the 5-phosphate of the next nucleotide. So it can add if there isn't a free 3' OH group available, but this problem is solved with the help of a primer which provides a free 3' OH end. Because DNA polymerase III can only extend the 5' to 3' direction, and because DNA double helix is anti-parallel, the DNA strands have opposing orientations. Only one new DNA strand, the one that is complementary to the 3' to the 5' parental DNA strand, can be synthesized continuously towards the replication fork. This is known as the leading strand. The other strand, complementary to the 5' to 3' parental DNA, is extended away from the replication fork in small fragments known as the Okazaki fragments, also known as the lagging strand. As synthesis proceeds, the RNA primers are replaced by DNA. The primers are removed by the exonuclease activity of DNA polymerase I. The gap between the DNA fragments, the Okazaki fragments, are then sealed by DNA ligase, which helps the formation of phosphodiester bonds. And that is the end of DNA replication. So what happens if DNA polymerase I can no longer perform its 5' to 3' exonuclease function? DNA polymerase I possesses proofreading function, which lowers the error rate during DNA replication. During proofreading, DNA polymerase enzymes recognize any errors and replace the incorrectly inserted nucleotide so that replication can continue. If DNA polymerase can no longer perform this function, then the mistakes made in DNA replication cannot get repaired. Incorrectly paired nucleotides that still remain following the repair become permanent mutations and can cause a mutation like this, for example.